Thank you very much for coming along, everyone. Our RSE South webinar is on reproducibility today, and we've got two speakers. We've got Eilis from the University of Exeter, and we've got Alex from the Rosalind Franklin Institute. So Eilis is going to go first. We're going to break the mold a little bit today and do the Q&A after each of the talks. So if you have any questions while Eilis is talking, please just pop them in the chat and we will get to them after. You can also pop your mic on and ask questions after her talk's finished as well. Okay, Eilis, over to you. Thank you.
Thanks, Bliss. Yeah, so for those of you that don't know me, my name is Eilis Hannon. I am an EPSRC Research Software Engineering Fellow here at the University of Exeter, where I am based in the Complex Disease Epigenetics group. So I very much describe myself as an embedded RSE. And what that kind of means is that for a very long time now, I have been the programmer in the group that everybody comes to with their questions. It also means that I am the person who is there to kind of champion improving the software, improving our skills so that we can write more robust code. And I've been doing that for quite a long time through my involvement with the Software Sustainability Institute. And I'm more than happy to admit I've had a very mixed bag of success in convincing my colleagues to maybe adopt new methodologies. And in this talk, what I really wanted to do is share some of that perspective. And I think I have an insight now into how to effectively communicate with my colleagues what their motivations are. And I want to share that so that we can all benefit. I don't think I'm going to be saying anything particularly radical, but maybe connecting a few dots together that will ultimately enable us to be a bit more effective in how we engage with researchers outside of the kind of traditional RSE community. So I often think reproducibility has become a bit of a buzz, but it's really helpful to kind of take a step back and reflect on where it sits in the ecosystem of what we're trying to do. So we are in research-focused institutions, and research is about methodologies, a kind of a systematic way of finding answers to questions. And so what we do is we have hypotheses. We want to test those hypotheses. So we come up with ways of collecting data, gathering evidence. We then try and summarize that and disseminate that to the wider community. And then what we tend to do is refine our hypothesis, maybe adjust our theory, and then the cycle starts again. So we come up with a new set of evidence we want to collect, a new way of harmonizing that, and we share it further. It's important to notice that progress isn't always linear, but it's about building blocks of knowledge. It's also a kind of worldwide collaborative effort, and you'll have multiple groups working on the same problem, inevitably spawning slightly different ways of going about answering the same question. And this is really helpful because as researchers, we don't really like seeing something once. So if we gather some evidence that supports our hypothesis, we often don't believe it. And our first objective is to try to break it. And when you have this situation, and it can be helpful, therefore, that you've got different groups tackling the same question, because what we're looking for is this kind of convergence of evidence. But the downside of that is you can end up with a lot of different results, a mixed bag of results in the field, and it can be hard to synthesize into a single theory. But this is where we draw on our skills as researchers. We assess the quality of the studies. We try to we have in our mind what we think would be a rigorous and robust result, and we try to parse our way through the literature to find kind of the path of least resistance and think about how these results can be woven together into a theory that we can then start the cycle again and go off and test. We really like it as researchers when multiple studies converge on the same result. But as we're probably all very well aware, around 5 to 10 years ago, we started to an idea started to gather momentum, whereby there was a recognition that actually the vast majority of findings were not being validated. There are a lot of publications, there are a lot of studies in the literature where people had struggled to reproduce those results, and that was a challenge because it made it difficult to determine the direction of travel. It was potentially problematic because it might have suggested flaws in the methodological process. And so on this slide here, we've got a summary of some figures from a paper from an article in Nature, where they did a survey to researchers. So that's what's shown on the left. And they found that more than half of the researchers they surveyed did think that there was a problem with reproducibility in the field. And really interestingly, on the right-hand side is a graph of scientists' attempts to reproduce results. And you can see that it varies a little bit by field, but there are some quite high percentages of researchers who'd failed to reproduce results from someone else's study. And interestingly, almost as many researchers struggle to reproduce their own results, which is probably even more concerning. Now, whilst it's important that this does gather some attention, we often hear this phrase reproducibility crisis. Research is not reproducible, bottom line. And this narrative is really great for grabbing attention and getting it in people's kind of the forefront of their mind, and getting funders, journals to maybe change their processes to try to encourage research that is more reproducible. But it's a little bit problematic for researchers individually. It can be hard, you know, at the end of the day, we're all people, not to take this as a personal criticism about your work. And also it can become somewhat paralyzing, that if I'm now going to make my work open and people are going to start judging it by these reproducibility standards, then I'm going to get criticism and my work isn't going to stand up to scratch. And that's quite a hard barrier sometimes for people to overcome, particularly if you are not necessarily, you haven't necessarily been taught the tools to perform your work in a reproducible manner, or it's a mindset that's coming in at the end of the project as opposed to something that was kind of threaded throughout it. It's quite hard to at the end of a project to reintroduce reproducibility from the beginning. So one of the solutions to this crisis has been very much focused on code and software. And there's been a drive, therefore, for research in general to be underpinned by better quality code and software. And that's where we as RSEs come into this. Now, the presence of RSEs alone is not the solution to solving all the issues with reproducibility. Certainly, they can come in and kind of lead the way. They can be the kind of flagship bearers of better software for research. But as well as actually producing that reproducible research, one of the roles of RSEs is about upskilling our colleagues so that they become better programmers and that they're in a position to share the code that they develop in an effective way, and also to write code that is more reproducible from the start. But I think it's worth bearing in mind that these researchers that we're going to be working with come from a really broad spectrum of ability, and therefore their kind of background skills and knowledge is going to have an influence on what skills and what techniques we're going to be able to convince them they should start to adopt. It's also, when we talk about open code, we sometimes generally throw around terms like professional software development. And given that most research coders are in fact self-taught, they do not see themselves as software developers. And therefore they might start to think that a lot of what we're talking about doesn't apply to them. And they start finding, you know, if you go back to what I was saying before about how challenging it can be to make your research available and subject yourself to that criticism, you start wanting to try to make excuses for why this doesn't apply to you. And if you see yourself as a kind of a small, low-level coder who doesn't develop software, and the conversation is all about generating robust research software, you quickly start to convince yourself that you can ignore a lot of the precedent around you, or a lot of the guidance that you should be making your software openly available. And this, I think, comes down to a little bit of the fact that we have different variation in people who use code in their research. We have to remember that when we're thinking about how we're going to reach these individuals. So on the figure here, I tried to put together a kind of a spectrum of coding ability. And I think you can often detect where someone sits on this based on how they talk about how they write code. So, for example, there will be the real kind of people starting out on this journey who very much use code as a tool to get their research done. They don't particularly necessarily enjoy it. They know it's an essential part of what they're doing, and it's probably quite far from what they thought their PhD or their project might look like. And they very much don't think about what they produce as being a program or being a software. And so if the narrative is all about producing reproducible research software, then they can quite easily convince themselves that they're not doing anything particularly novel in this space, and therefore all this open code stuff doesn't really apply to them. I think though, once you start to use code more frequently in your research, you quickly do realize, you start to want to do a better job of it. You start to recognize where you can make efficiencies, and you start to talk a little bit more about writing scripts. You might call them small programs, but your terminology kind of moves away from just thinking about using code to kind of writing code to do a particular task. And you recognize that you need to write a script because you want to document what you're doing. Occasionally, you might be doing something novel, and you're also interested, you know that you're going to potentially have to share this or with your colleagues, or you might be reusing it yourself. I think the shift from kind of using code to writing scripts is quite natural, and most programmers probably get to that point. But then I think there's a really big jump going from that to thinking about developing software, where you are thinking kind of more conceptually about how you share your functionality with other people in a way that's generalizable. And is, you're starting to think about situations where people who aren't necessarily doing the nitty-gritty of what you're doing might use it. I think there's a really big jump from the middle of this graph to the far right. And it's important to think about where people sit on this spectrum, because how someone views their own individual competency is likely going to influence how much they think open code applies to them, and how what aspects of open code is most relevant for them. Now, we don't all necessarily need to know how to develop robust software in order to produce reproducible research code. So this, theoretically, anybody on this spectrum can be doing things to make their code more reproducible, and certainly can go about developing it in a way that they can share it. But it's critical that when we think about trying to upskill our other researchers, we recognize that not everyone's on the far left or far right of this diagram, and not everyone wants to or is going to get to the far right, and that potentially we adjust our expectations to meet the researchers where they are. Trying to push them too far, in my experience, often gets met with resistance and then completely disengaging from the process. We need to think about kind of iterative, stepwise things we can encourage them to do that start to edge them along this diagram here. So it's worth acknowledging at this point that definitions are important, and not only does everybody have maybe a different underlying skill set, what they're trying to achieve when it comes to reproducible research might differ as well. So you've probably seen variations of this image about the difference between reproducible research and replicable research. So reproducible is about getting the same results with the same data set in different hands. So if I provide you with a data set, you can, and I provide you with my analysis, you can get from A to B and come out with the same output. Whereas replicable is about repeating the analysis, so potentially on different data sets. So it's more along the lines of kind of independent validation. And different projects will have different requirements in terms of whether they need to be reproducible, which is arguably the minimum standard for kind of all research, whereas some studies will also have the expectation that they are replicable as well. I want to acknowledge that there is some variation in the field as to the precise definition of these two words, but this is how I'm going to refer to them today. And replicability is particularly important if you're talking about a project where you are developing a new methodology that you want to share. And what I'm going to do in the rest of my talk is kind of showcase how situations where the needs of making the code open was slightly different, and how we went about doing that in that way. So before I talk about the specific case studies, I just wanted to set the groundwork for the kind of research that I'm involved in. So I'm part of the complex disease epigenetics group. And so what we're interested in is profiling the epigenome and looking for associations with different diseases and phenotypes. And so if you're not familiar with the epigenome, it's basically a set of modifications to DNA that controls gene activity. So whilst your genome is the same in every cell in your body, your epigenome differs in different cells because that's what drives different genes to be turned on and off, and those different gene activity is what drives cells to look and function differently. It means when we're looking at association studies, so if we're looking for epigenetic variation to be associated with a particular disease, we're looking at a scenario that's quite similar to traditional epidemiology. And for anyone familiar with epidemiology, study design here is really critical to how you interpret the results. And there's a huge number of confounders that could leave rise to false positives, which we need to control for in our analysis. So the analysis tool that we use is fairly typical, and it's a kind of regression model, but there might be some fine tuning in the exact model that you apply, depending on the study design, the phenotype that you're interested in. And so whilst everyone's using linear regression, the kind of specification of the model is likely to change. So the first case is indeed that it's an epigenetic epidemiology study. And so on this slide here, we've got a little schematic. This is some DNA where it's kind of wound around these histones, and these red lollipops highlight different epigenetic modifications that we've quantified. So the data that we get out of this is essentially a huge matrix where each row represents a different position in the genome, and the columns represent different individuals. And there'll be some phenotype or outcome variable that we can use to capture variation across our sample. So here we've got different colours to distinguish cases from controls. When we come to writing our code to analyse this data, what we want to do is essentially a statistical analysis comparing these two groups. And we want to fit the same regression analysis to every position in the genome. So, which is the same, essentially the same as saying fit the same regression model to every row in our matrix. This is a highly repetitive task. It would have been completely infeasible before we had computers, but it's not a particularly complex methodology. It can be done with kind of base functions in R, for example. So our script isn't particularly complicated to do this, but we do need a script and we do need programming to complete this task. Now, in the case of making this code available, we have to give some thought to what our objective is to decide the best mechanism for doing that. And the key crux here is that making our code available is about us being transparent in the methodology that we used. So we want to clarify the exact regression model that we used. And the purpose of making our code available here is such that if somebody had our data set, they could rerun our analysis and get exactly the same results that we published in our manuscript. So our objective here is reproducibility of these specific results. And the code that we want to make available is tied specifically to a set of analyses from a specific publication. So what that means is that annotation of the code is important because it's likely to be interpreted in the context of the method section of this paper. Some lightweight documentation is going to be helpful, but what we really need to ensure is that what we're preserving here is the final version that was used in the analysis. So we don't necessarily need kind of detailed version control of how the scripts changed over time. The critical bit that people are interested in for the purposes of reproducing our results is that final version, and that is the thing that then needs to be preserved throughout time. And as we're putting this available, and it's possible people might want to take what we're doing and develop it for their own purposes, a license would be important as well. So this isn't going to be a software product. It's very much a documentation of the code that we ran to perform these analyses. And so the mechanism for doing this was to put it in a GitHub repository, because GitHub is a place where people know to look for these things. It's an accepted route by the journal that we published in to host these data. But this isn't a repository that's going to have a huge backlog of commits, as you can see here. we're not going to have a huge number of releases. In essence, this data, these scripts are going to be put on this repository and probably and theoretically never touched again, but they need to stay there for the purposes of preserving the methodology, and such that anyone reading on publication can map between what we present there and what we put here. So we have quite a lightweight README that very much links this back to the publication, but the repository itself is quite light touch. In this situation, diving in and talking about kind of version control in its really kind of intense state is not necessarily appropriate for the developer of this project. It's more about thinking about how a repository like GitHub can be used to share your code and to provide additional information that enables somebody to understand what it's there for. The second case study goes a bit further than that, and here we're talking about sharing a methodology that we might have developed. So one of the things that epigenetic data is quite frequently used to do is to estimate the cellular proportions of a bulk tissue. So I mentioned at the beginning that different cell types have different epigenetic profiles. And when we profile a piece of bulk tissue, so for example, some brain tissue, that tissue consists of multiple different cell types. And therefore, the profile that we generate from brain is essentially an aggregation of the cell types within it. This can be problematic because it means if the ratio of the cell types differs between the thing that you're interested in, then that could induce false positives. So in a typical experiment, we've observed the brain profile on the left, and we haven't observed what's on the right-hand side of this equation, but we know that we want these proportions to include as covariates in our analysis. It's standard in the field that if you don't include these, you will be criticised and probably struggle to publish what you've done. However, because we can represent this as a mathematical equation, if we have some of the unknowns on the right-hand side, we can start to estimate the proportions. So we know the reference profile of a neuron, we know the reference profile of a glial, we can substitute these in and derive estimates of the proportions when we've got brain data, and we can then take those forward for our covariates. This is a very well-established and commonly used methodology. The challenge, though, is that it's ultimately a mathematical formula. You put numbers in, you get numbers out. And because of the way it's designed, the numbers you get back will look like proportions. But that's not to say that this model is appropriate for the application that you've used it in. So people often apply these because they know that they're going to struggle to publish their work without these covariates. They just use these algorithms because that's the standard, and don't necessarily consider whether they're the most appropriate for the scenario that they are working in, and partly because they also don't have a mechanism to quantify whether the model is appropriate for the data set they want to apply it in. So that was a methodological gap that we wanted to address. And so we did that. I won't explain the nuances of it, but we came up with this mathematical algorithm that developed a score called SETIGO, which enables you to quantify the accuracy of the deconvolution from the previous slide. And we can show that this works because we can do a kind of nonsense situation where if you're trying to deconvolute blood cell types, you would expect that to work in blood tissues, but to not work in tissues from other organs of the body. So here we've got our error metric on the y-axis, and the pink violins here represent sensible applications of the model. So we've predicted blood cell types in blood samples, and the scores are lower. And then on the right-hand side, you've used the same algorithm, but in tissues that are not blood. So we've got buccal, we've got nasal, we've got a range of brain regions, and we've also got pancreas and the liver, and we can see our error metric is higher. So having proposed and validated this method, we now want to share this as something for the wider research community to use. So in this situation, we now care about replicability as well as reproducibility. And so the way we go about sharing this code changes because the needs of how we want to do that and what we want to achieve by making it open have changed as well. So in this situation, we've developed an R package for people to use. And the R package is introduced by the publication, but it's not tied so closely with it. It's more likely to evolve over time, and therefore the way we go about sharing it and changes from that first use case. So documentation is important, but also important here is the dependencies which you need to set up to enable the functions to work. Version control becomes more relevant in this case because the package is likely to evolve over time, and we want to be able to document that to people who might come and use it at different points in that process. So this is an example where we are anticipating people will want to reuse our code for other situations, and therefore the way that we make it available differs from the situation where we just want to document how we ran the code at a point in time. So release management is important, even though when we started out on this, I didn't imagine us adding any additional functionality. It's amazing once you've set a framework up for sharing methodologies, how easy, how you do start to think of extras and extensions that you would like to incorporate, and this has inevitably been updated and had a couple of releases. And of course, the license is really critical here, particularly if you want to enable people to build on what you've done and enabling them to do it. In fact, in this project, we actually had 2 GitHub repositories, because not only did we want to share the methodology such that other people could use it, so there was one GitHub repository related to replication. We also wanted to have either a GitHub repository or a set of scripts that documented how we did the analyses and the manuscripts that validated the method. So in fact, we had 2 parallel objectives here. We had both reproducibility of the findings in the manuscript, which had one set of requirements, and we also had the replication of the methodology we were proposing to the field. So this project here, because we were trying to share our method, if you look at the GitHub page for this, it looks a lot more like a kind of software product, because it's much more in that vein. We've got a much more active repository here, and version control is a much more relevant concept in this space. So we've got it set up like a kind of standard R package. We've got a much more detailed README that includes more documentation on how to use the scripts. We've also taken advantage of the wiki pages to provide tutorials to really enable users to take our methodology and apply it. Because in this situation where we were much more motivated and we saw the value of the methodology being reused in other situations, and indeed that was the purpose of the project there. And so unlike the first project, the needs here have differed, and therefore the skills and the way we go about it have changed. So I think that's hopefully about my 15 minutes, and I just wanted to finish by reflecting that for many researchers, sharing their code the first time is a really hard and scary boundary to overcome, and therefore we need to be realistic in how what expectations we set to help them overcome that. We need to try to find a way, therefore, of meeting researchers where they feel comfortable, understand the requirements of what they're trying to achieve by open code, and not kind of push them before they can swim. Think about kind of meaningful small changes they can make that actually serve their purposes. So ultimately encouraging kind of stepwise change in coding behaviours, and recognising that not necessarily all research code needs to be packaged as software or kind of promoted to the same standards to be open, and kind of on a case-by-case basis determine what it requires and what kind of open means in that situation. And there we go. So that's me. Thank you.
Thanks, Eilis. We have some time for questions. I think Tom has popped a question in the chat.
Yeah, let me just pull that back up.
And we've also got a hand up.
Well, more of a...
Shall I start with Tom?
Yeah,
so
sure.
Tom's question is, how do you think the level of software testing varies between reproducibility and replicability? In reality, it probably varies dramatically. I imagine in reproducibility, you have little to none testing. And I guess it would be a very bespoke, because you're designing software in that situation often for one use case. you don't have such a perspective on the generalizability. And as long as it works for the use case that you're specifically interested in that point in time, you don't think much more broadly than that. In replicability, you're probably a lot more conscious that if other people are going to use this in context outside of what I'm trying to do, you want to understand that a little bit more. Or if you don't understand that, you're going to get questions when people try to use it and it behaves funnily. Yeah. Hope that answers your question, Tom. Robert, did you want to come in?
Yes, thank you.
Hi. Yeah, so really good talk. I really liked the model of sort of coders, scripters, software as largely because it chimes with my experience, and that's why I like it. But I think, yeah, I think that's good. Do you feel that version control with Git and GitHub is an unrealistically high expectation for people at the coder end of that spectrum from what you've seen?
I think we need to evaluate what aspects of GitHub and version control we teach first. I think introducing it as a platform for sharing code is more in line with what those users are going to use it for. Because I think throwing version control often starts as this tool for collaboration, and then we say, oh, and then there's this really nice web interface GitHub you can use to make it a little bit easier. Whereas in my reality, most of my colleagues use GitHub to post their scripts and preserve them in time. And if they do version control, it's only ever with themselves. And so therefore, it's arguably how you approach it is a slightly different task. For example, you very rarely have merging of branches. I feel like there's maybe a better entry level if we start off by talking about the different levels of sharing code. It's just in my experience. I think a lot of my colleagues have successfully put their scripts on GitHub and left them there. I don't know how many of them successfully use GitHub to collaborate with another colleague or even really collaborate with themselves in the future. That there's definitely a barrier there that they haven't been able to get over yet.
So I was thinking more about the challenge for an individual researcher of just keeping track of what version of code they use today and yesterday, rather than in a sort of reproducibility sharing context.
So should we be encouraging people to use version control for that purpose?
Yeah.
I think we should. We should definitely be trying to encourage them. I think we just have to recognise that the carrot for them might be different.
Mm-hmm.
The carrot isn't necessarily about these big multi-person projects. It's about you. It's another way of documenting how your process evolves over time.
Thank you.
Yeah.
I realised I'm muted. Thank you very much, Eilis. I think in the interest of time, we will move on to Alex's talk. And I will let you kick off, Alex.
Okay, thank you. I'll just share slides. So hopefully you've all got the full screen view now and can hear me.
Yeah,
great.
We do.
Okay. So hi everyone, my name's Alex Lubbock. I'm a senior RSE at the Rosalind Franklin Institute, and today I'm going to be talking about automated metadata capture in Python using a library called Microbench. So this is a library I wrote actually in my previous position where I was working as a research assistant professor in a computational biology lab at Vanderbilt in the US. But it's kind of got broader applications than just computational biology. It could be used for other types of computational workflows too. So I'm excited to share it with you and then hopefully get some feedback. And it's open source software, so it'd be great to get some people using it and to get some feedback on it. Okay, so aspects of reproducibility. So Eilis sort of gave an excellent introduction to this, so I won't try and recover too much of the ground, but essentially when we're talking about reproducibility, well, one definition that I'm using here is boils down to, can we match the conditions from an earlier code run, right? We expect the computer to be deterministic, we hope it is. So if we can match all those conditions, we should get the same results out. But conditions can include many different things. So the software that you're running, the version of that software, its inputs, so what files you're using as inputs to some computational pipeline, command line interface arguments, the dependencies of the software, so versions of Python packages, for example, other installed software, so things that you might not realize are actually an explicit dependency, but actually your software is using somewhere down the chain that might have an impact on the reproducibility of your code. Less likely, but still possible, things like the version of the operating system that you're running, system library versions and things like that can, in sort of some edge cases, have an effect. Hardware, so Intel, AMD versus ARM CPUs. In the Python world, we hope things are cross-platform, and they usually are, but bugs can still creep in that lead to differences. Downloading files, this is kind of an interesting one, right? Because this is if you're relying on some external file being available and downloading it as part of your pipeline or some external API, and then suddenly that stops being available due to lack of funding or the server crashing or something like that, and you get a 404 error, then maybe your pipeline stops being able to run at all. Or even more insidiously, maybe someone changes a file on a remote server and you might not even realize that file has changed and suddenly you get different results. So one way around that might be to check the checksum of the file that you're downloading when relying on external files. And stochastic code elements. So in some domains, like when you're doing computational simulations using Markov chain Monte Carlo, for example, there are random number generators involved. So depending on the kind of level of reproducibility that you're expecting, you might need to include sort of fixed random number seeds for that. So all these different things can play into whether some piece of code is reproducible or not, or some analysis is reproducible. So some scenarios, I'm sure we've probably run into a few of these, many of us. So if you're working some code associated with a publication that someone else has published, you download it, you struggle to get the code to run. Maybe there's not enough instructions or information on the requirements. Maybe they wrote it on a ARM MacBook and it won't run on your Intel Windows machine, for example. Maybe you can get it to run, but you get the results to differ and you're not really sure why. Of course, you can try to contact the authors, and most authors are helpful in my experience, but sometimes you might not be able to get hold of them. The person who wrote the code might have left the lab, so there isn't always a route to resolve the issue there. So it's better if we can try and make things as reproducible as possible from the start. Another scenario, developing code across multiple platforms. So if you're working on your laptop and for development and then running some code on a Sloan cluster, for example, maybe there's some differences in those environments, like the CPU architecture. There's a module share on the cluster which you haven't got on your laptop. There could be these differences that have an effect on reproducibility. And an annoying one that I've certainly run into, and I'm sure many other people have, is that you run some code, you write some code, and then you come back to it 6 months later or sometime later, and you're struggling to reproduce what you did before, and you're wondering, well, what's changed? And if you're using Git or version control, then you should be able to look back as the code as it was then, but you might not even know the exact version of Git, the Git version that you were using at the time, right? So the hash of the code version. So you know that the version was at least 6 months old from that point, right? If you come 6 months later, but you don't know the exact version that was used necessarily, unless you explicitly note that down for an analysis. So you don't kind of have all this context about the environment. So part of, I think one tool to help this would be metadata capture. But just to say that this sort of sits among a broader ecosystem of tools and best practices for reproducibility. So versioning data sets, having version control for code, having requirements files, virtual environments, these kind of things can help for making sure you're running the same versions of software dependencies. And then kind of going further down the increasing the reproducibility, but also adding sort of a burden on the software author, is doing things like containers, which can help to add reproducibility by making more of the operating system libraries and things like that reproducible. So you have the same versions of those. And then sort of there's other techniques that sort of I think would probably be considered perhaps a bit extreme and overly burdensome, like using full virtual machines and emulation. But if you really strongly want something to be reproducible, then those kind of techniques have their place. And then for sort of another kind of important thing to mention is testing, right? So you could have integration tests and end-to-end tests that ensure that the code, and you can integrate those with continuous integration, something like GitHub Actions, to make sure the whole code pipeline runs through and gives the results that you expect. Whether that's feasible or not depends on the type of code. If you've got something that relies on GPUs, for example, you'd have to do your own custom GitLab or GitHub runner for that. So it can become basically difficult to the point where it's not commonly done for people to do end-to-end tests for certain types of code, and particularly computationally intensive code, where it's probably not feasible just to run it on GitHub Actions end to end. So these things are a selection of things that can be used, and the more of them that we can use, the better. But there are sort of limits to what people have time to do and what people have the skill sets to do as well. So okay, so metadata capture. And kind of giving a definition for this, because this is what the library I'm going to introduce today is focused on. So if you imagine you have some computational pipeline or a script, in the simplest case, where it takes some input files and arguments, you're running the program, getting some results out. Metadata capture is capturing aspects of that environment for you to record, so you have a record of it for later. So this could be things like the input file names that you used in the arguments, the software architecture, the version of the GPU, for example, the model and make and things like that, versions of the NumPy in this case, but other Python packages. All these things could be considered metadata tags, and then you store them in some place, right? This, in the simplest case, this could just be a file. You could put this in a central database. So the library I'll introduce supports both of these use cases, but this is kind of the central concept of capturing these metadata so that you know the environment of some code run. And kind of put this a bit more into context, final slide before I talk about the tool itself. You can go from sort of being not reproducible at all. If you don't have the code and the data to do the analysis, then of course you can't reproduce it. But if you just publish code and data and there's no documentation or information on how to reproduce it, it can be pretty difficult, right? So there's an element of maybe you're fighting compiler errors, you're trying to work out what platform this was written for, Intel versus ARM, that kind of thing. But this depends on an element of luck and skill in being able to sort of run through that debugging process. Better documentation is really going to help with that from the user's perspective. And then, as a gold standard sort of best practices, we might add runtime environment replication, making the installation actually easy to do, having validation tests and so on. These things are not always done because they take a lot of time and effort to set up. So that's where I think metadata capture can really help, is that it is a fairly easy thing to set up. And even if you don't have a completely easy to reproduce environment, at least you know what the original environment was, so that you can work towards reproducing that later if needed. Okay, so on to Microbench and how it works. So for those of you who might not be as familiar with Python and the concept of decorators, here's an example of what they do. It's very simply just a wrapper for another function. So one function that wraps around another. So we have this say we function, which just prints the word we, and then we have this decorator function, which basically can wrap any other function. And then we apply it using this little syntactic sugar of this @ symbol in the name of the function, which means we can apply this my decorator is then a wrapper around say we. So what happens is when you call this say we function, the wrapper runs this code before, runs the original function, then runs some code after. So it's a way of making one function surround another like this as a wrapper. Okay, so onto Microbench and how it works. So the way that you use it is to define a benchmark suite, as we're calling it. So this is just a very small Python class where you give it a name, set up where you want to save the metadata, and then basically specify what metadata you want to capture. So this could be a variety of different things, from package versions, environment variables, information on the host computer. I'll come up a future slide, in a couple of slides time, I'll show you all the different types of things you can capture. And then when you want to use this benchmark suite, you just decorate the function that you want to analyze, and then this metadata gets captured automatically when this function is run using this wrapper functionality. And then from those, from the metadata file, you can, it also captures as a bonus, it captures benchmarking data. So you get the runtime of this function and how long it takes to run. So when we were developing this package, one of the things that people wanted to do was benchmarking of their code to work out how long various functions would take to run. And so this is kind of a sweetener, right? As well as reproducibility, you can also do benchmarking at the same time. So this is one of sort of the incentives that we found useful to get people to actually start using this package. This is published as a bioinformatics application note. It was published last year. I'll include the references and the download links for the code and everything at the end. Just to go a little bit more into detail about how to use it. So you have these benchmark suites. You define the metadata you want to capture using so-called mix-ins. These are small Python classes. So Python has multiple inheritance. You can put a list of as many of these classes that you want to inherit from at the end, so that you can capture whatever metadata you need to. You can add your own custom ones as well if you like to. The Microbench library is lightweight. There's no dependencies beyond the Python standard library, although some of the mix-ins do require some extra packages if they're needed for extra functionality. As I mentioned, it's useful for both reproducibility and benchmarking. You can store the results in a file or in a Redis database, and we could potentially add other centralized metadata stores if there was interest in doing that. And the metadata format is just in a JSON lines format, so you don't need Microbench itself to do any analysis. It's all in an open format, so it's easy to come back to later. So the types of metadata that you can capture and the things that we found that were useful. By default, we capture the start time and finish time. So from that, you can get the runtime of the function name, and then you can specify these different mix-ins depending on what you want to capture. So you can have the global packages. These are all the Python packages that have been loaded into the current environment. The installed packages, so this is everything that's available for import, so the package names and their versions. Conda packages, so if you're using Conda, you can capture everything in the current Conda environment and all those package versions for archiving. The function call, so the arguments and the keyword arguments used, the return value of the function, the Python version that was used, and the executable location, so it helps to indicate if there's an active virtual environment, information on the host, CPU cores, RAM, information on GPUs, and line profiler, which I'll come to next, and this is useful if you want to do more on the benchmarking side. You can just include this line profiler mix in, and then this is my function, and it's deliberately inefficient. It's adding numbers one by one in this loop, which is a terrible way to do it. But if you've not used the line profiler library before, this is the kind of output that you get from it. And essentially, you can see from this % time that we're spending all of our time in this loop here. So if this was a less contrived and more complicated example, it gives you a nice way of seeing, oh, this part of my code is really inefficient. And you can kind of capture this on the fly and kind of archive it for later. Last final feature I'm going to highlight of the library is this feature of telemetry support. So this lets you capture metadata ongoing using a background thread that's included in your Python process. So in this case, we want to capture memory usage. So maybe we think we've got something that's using up a lot of memory, and we want to see how that memory usage grows over time. So we can specify a telemetry interval of 90 seconds, and then this function is essentially like a callback that captures. You can take anything from this process and capture it and store it as metadata. So in this case, we're capturing the memory usage every 90 seconds as part of our benchmark. Okay, so that's the Microbench library, a quick whistle-stop tour. So quickly summarize, the metadata capture is kind of one part. It's relatively simple to implement, so and it can help also, as a bonus, you kind of get benchmarking data, which can help to persuade people to adopt it. And it's extensible, so you can add additional mix-ins and classes if there's additional metadata types that you wanted to capture. So I'll just finish up by acknowledging Carlos Lopez, who I co-wrote this package with, the funding bodies, documentation and codes available on GitHub. It's open source, MIT license, I think. There's the manuscript, and there's a website for more about me. So I'll finish there, and thank you very much.
Thank you, Alex. That was great. Does anybody have any questions for Alex? Yes, we've got one in the chat.
Okay, so the question is it the second one? I'm just trying to see.
Yeah, could you say something about the performance penalty/overhead of using Microbench? Can you turn it off automatically?
So turning it off, yes. I mean, you can just comment out the at line of the decorator if you didn't want to use it at all. So a quick one-line toggle if you don't want to capture the metadata. The performance overhead, there's actually a thing on the README on the website about that. It depends on the mix-ins you're using. So if you want to capture things like the GPU information, that invokes NVIDIA SMI, which takes maybe up to a second to run. So yes, it really depends on the mix-ins you're using. If you're just doing the very simple default ones are capturing the start, finish time, and function name, then there's barely any impact at all. But yeah, so there's not too much impact depending on which of these you want to use. But if things like the line profiler and NVIDIA SMI are a little bit heavier weight, so you probably only want to invoke those when necessary. You probably want to do them on every function invocation if you're doing that in a tight loop, but doing it once per program is usually fine.
Thank you. We've got another question. Any thoughts about how a system like Nix/OS might fit in here if you are familiar with it?
I am not, I'm afraid. So I'll take a look at that. But if there's anything specific that is worth how that's related, because I'm not familiar with it at all, I'm afraid. So
it's no problem. Oh, yeah, go on, Nicholas.
Yeah, so that was my question. Just to give a broad overview, I think that when you're looking at capturing metadata, that isn't sort of your classical benchmarking, like how long does a function spend here and there, and how much RAM is used. All the other stuff seems to be like about capturing the environment. And Nix OS and the Nix system is sort of like a package manager for your entire operating system, where you define the entire environment and specific parts of the environment that you run software in a text file, essentially. And I think, as you say, it's not sensible to to enforce this kind of system on every user, to say this is how you approach reproducibility. It's a place for each different piece of software, but it does seem like there's a big overlap there, and that would be an interesting way to build with this in mind, instead of going backwards and saying, well, once we built it, how do we capture the metadata?
Yeah, so I think it does sound interesting. I think I might have come across it briefly before, but I've not used it. I think it kind of fits in with a lot of these. So you have sort of things like Conda and containers as well that can provide some level of standardization for environments. I think the difference with the metadata capture side of things is that you can, by capturing these metadata, it's relatively simple to do. Whereas I think a lot of people are quite daunted by, say, writing a complete Docker environment, and maybe they're not using that Docker environment on their laptop, they're only using it in different places. So by capturing this metadata, you can kind of look for these differences automatically, right? And you can output them to a JSON file, And then we found that you can just use a JSON diff tool to look for differences. So you know, if you've got like say you've got like a pip requirements file, but you haven't specified exhaustively all the dependencies, and there are dependencies of the dependencies, then those are the kinds of things that you can pick up as differences by capturing that metadata. So I think they're kind of complementary approaches to do both the metadata capture and to have rigorously specified environments.
Matthew, did you want to come in on this?
Yes, yeah, I sort of had a similar question, and now thinking about it, so I guess the risk with this sort of Microbench approach is obviously the user has to sort of decide what metadata is actually captured, and therefore is there not quite a risk of important metadata being missed, and if so, like, is there sort of standard templates of like the metadata you should be capturing?
Yeah, I think that's a great question. At the moment, we kind of went for the flexibility approach of leaving it down to the user what they want to capture. Because as the earlier question alluded to, some of the things have a slight performance penalty to do them. So you have to be careful if you're capturing them within a tight loop of a function being called lots of times. But I think having standardized benchmark suites of saying, or kind of metadata lists that you want to capture would be a great idea. I don't know what level you would do those on, whether they would be sort of a community-wide metadata standard, or if individual institutions would specify those, but having kind of minimal standards for reproducibility and the information required for that would be a great thing to sort of pre-specify. And then that way we could have these as available imports within Microbench. You could just import, you know, RSE level 1 benchmark suite or something like that for reproducibility and have that available.
Great. Well, we're perfectly on time. So thank you, Eilish. I think she's dropped off the call now. Thank you, Alex. And thank you everyone for joining us. I'm gonna ask everybody if anybody who's here would like to speak at a future webinar, please do get in touch with me or any of the other people in the webinar organising committee, and I hope to see you next time. Thank you very much.